Yeah, well, thank you for the invitation. My name is Rômulo. I'm working with Richard Buggs, and yesterday Richard said I'll be speaking about acute oak decline. As you can see, we are one species short of that goal, 5 if you count the beetle and bacteria. But I'm pretty sure, absolutely certain, that birch is your favorite tree, and you're extremely interested in knowing whether silver birch is adapted to the environment in Britain and what makes a tree grow well. So because you want to know that, we went out and we phenotyped and sequenced the whole genome of 2,200 trees. You're welcome. These trees mostly come from a Provenance trial planted in the 90s. The trials are 3 in Wales, England, and Scotland, and we have 29 Provenances, but we also have plus trees, around 150 from the north of England and Scotland. They're in the Glencore's archive. Of course, this is a huge effort, and it was only made possible because of CFP funding and our collaborators, Forest Research and the Future Tree Trust, who did all the phenotyping and gave access and helped us out. So it's a joint effort, and we also have researchers in Sweden involved as well. Yes, so these are the sites. As you can see, they're pretty different. Particularly Durham in Scotland is a much harsher site. And if you take the environmental conditions of the source, location of the provenances and the sites, and you summarize that in 2 axes in a PCA, you can see that Durham is quite far away from the envelope. So it's a very different site from what these populations are used to. This is going to be important later. So FR, with some key researchers, they've collected a bunch of phenotypes related to growth, you know, your classics, DBH, wood density, height, water use efficiency through carbon assimilation, the health of these trees, and bark thickness as well. So the first thing we did was to look at the phenotypes themselves. Oh, and by the way, this paper, it's being submitted to a journal, and there is a preprint. If you scan that code, it'll take you to the paper. There's more results that I will not be able to discuss here. So first of all, we just looked at the phenotypes. On your x-axis is just the latitude of the provenance, the source provenance, and the colors are the site. So the first thing you can see, there's a clear side effect on most of the phenotypes. So for example, DBH, trees tend to be larger when they're planted in Drummond, but there is a general trend negative trend in latitude, which means that provenances from the north, no matter where you plant them, even when they're planted on the north, they tend to grow less, okay? And they were quite unhealthy in one particular site, Landover. So the next thing we did was to look at the genetics. We wanted to know the genetic architecture of these traits, so you could have a trait that is controlled by 1 or 2 genes with very strong effects, or it could be a polygenic trait like Danny was speaking about. So this is a Manhattan plot. In the y-axis, x-axis, you have the chromosome position. Each dot is a variant in the genome, like Danny was showing you. If there is a strong effect of a couple genes, you would expect to see very big peaks, because it's the p-value, it's minus and log, so the taller the peak, the stronger the association. I'm just showing 2 traits, but usually what we found was not a strong association at all, or a couple SNPs that were associated with it, which suggests that either these traits are plastic, there's no genetic basis, or they are polygenic. There are many, many genes involved, and they have very small effect. Because of the previous slide, it seems that genetics is probably the culprit here, because, you know, these trees are consistently growing a certain way no matter the environment, right? So we calculated the heritability using the whole genome. Now it's not just individual variants. And as you can see, a lot of these traits have pretty high or moderate heritability, like 60% of the variation in height is explained by the whole variation in genetics. So it suggests that these traits are polygenic, right? They're controlled by many genes, small effect. So next we did genomic prediction. So the idea is you train your model on the genomic data and you attempt to predict the phenotype of, let's say, 10% of your set, and you rotate those sets until you test it every one. And we have moderate accuracy from 0.4 to 5. Part of this, I don't have time to speak about it, but there's a lot of genetic noise, background noise, because of the latitude gradient. There's a lot of historical neutral structure in these populations. But this is good enough to speed up, let's say, a breeding program if you wanted to. And this is the number of GWAS hits. As you can see, there are not very many. The next thing we did, we used the models that we trained on the provenance trees, and then we attempted to, we didn't have the phenotypes of the plus trees, but we attempted to predict the breeding values of those trees, the genomic estimated breeding values, and compare to trees from provenance at similar latitudes from where those plus trees were collected. The effects are not pretty strong, but the plus trees are the purple. As you can see, for example, for height, there is that classic decrease in the trait with latitude, but the plus tree slope is less steep. So it seems to suggest that the breeding values of the plus trees is a bit higher for a few traits in high latitudes compared to random trees, which seems to suggest that those plus trees were well selected, although the effect is quite small, okay? It's not super big. So next, Phoebe Swift. Now she's a PhD student with the ASH project, but she was an intern and then a research assistant in this project, all thanks to the CFP again. She ran models like the GWAS that I've shown you for the genetics and traits, but now we're looking at environmental variables just to see whether there is an association between genetics and environment, which is a sign of local adaptation. So she looked at a bunch of different environmental variables, temperature, frost, and contrary to what I found with GWAS, she actually found very, very many loci associated with environment, particularly temperature, precipitation, frost, and wind speed. So that seems to suggest there is some local adaptation. I tried to get more funding for this project contacting Taylor Swift, but it turns out Phoebe and Taylor are not related, in which case I didn't manage to achieve that goal. Next, what we did, which is quite exciting, we did what we call genomic offset models or allele shift models. We got those NIPs that Phoebe found, they're associated with the environment, and we attempted to come up with a model of adaptation. So you try to predict in a certain environment what would be the ideal allele frequency for those NIPs. And you see whether the trees are locally adapted to the trial sites they are planted. So you take the mean allele shift, the higher the shift, it means that those trees are more maladapted and they will need a higher shift to get to the optimum, right? And as you can see, Drummond has a much higher allele shift requirement, which would suggest that the trees that are planted there, they are maladapted, right, compared to the other two sites. The exciting thing is that they're still surviving pretty well. In fact, mortality rate was higher in Landover than in Drummond. And finally, we actually predicted, using the climate change worst case scenario for the source population. So how will be the environment in 100 years' time, worst case scenario for the source populations. And we checked how much allele shift would be required for the local populations to be adapted to that future climate. And it turns out that actually the shift would be lower than it is for Drummond, suggesting that perhaps Drummond is even harsher than the worst case scenario for climate change. But the fact that the trees are surviving there, it's good news. Of course, these models are not very good when you're trying to predict things outside the envelope, but if you take it at face value, that suggests that tree may survive if things go pretty sour, which matches what they're talking about epigenetics in the previous session, right? So the take-home message is, so the southern provenances tend to grow more no matter where they're planted. So if all you care about is timber, although the recommendations today is to always plant local, if all you care about is size, then perhaps planting from this, using provenance from the south would be better. However, if what you want is conservation, as we showed, there is local adaptation, so you'd be good to preserve the plants from the north because they're locally adapted to their environment, right? So it depends where your goal is. The GWAS, the Manhattan plots that I showed, and the heritability suggest that these traits are mostly polygenic. They're controlled by many genes with small effects, so they could be used for genomic prediction could be used for breeding programs in the UK to speed up the cycle. Plus trees tend to perform better than the trees that randomly sample, as I said, so it's a good thing, and so you could source from those plus trees to complement the planting in the north if you wanted to, but as I said, the effect is quite small, and progeny trials would probably be needed to confirm that superiority. I think Future Tree Trust is already doing that, so they could confirm those findings for us. And finally, Provenances predict to be least suited to a harsh environment, in this case, Drummond, are performing well. That could either mean that the models do not work very well when what you're trying to predict is very, very outside the environment that these trees are exposed to, and this has been shown in the literature before, but it could also mean that, you know, these trees may be doing okay despite the increase in temperature in the future, but more evidence would be needed for that conclusion to be sound. So as I said, it's a joint effort involving many, many people, and I'd like to thank the funders and all our collaborations. So thank you very much.